Hello dear students, welcome back to GDC. In this video, we are going to discuss about one of the important concepts of carbohydrate, that is identification test for carbohydrate. As you know, there are many tests are there in the carbohydrate and it is always very difficult to remember this test, which test is for the which carbohydrate, for what purpose it is used, what color it forms, what are the reagent that is used. It is very difficult to remember. In this video, I will try to share some important mnemonics tricks to remember what are the tests that is used for the different carbohydrates. Okay, let's begin with the class. So as you know, carbohydrates, if you'll see the classification of the carbohydrate, so carbohydrates are broadly classified into three major classes. So last class we have discussed. First one is your monosaccharides. Second is your oligosaccharide and your polysaccharide. Okay. So monosaccharides are those which are consisting of single carbohydrates, also single sugar units. Then oligosaccharides are again of two types, either they are disaccharide or trisaccharide. Polysaccharides are again two types, homopolysaccharide or heteropolysaccharide. There are different tests which is used for the identification of carbohydrate. There is a simple mnemonics for you can remember that the mnemonic is 8 important tests about which we are going to discuss today. So please see, so the mnemonic is bomb is fall secretly or safely, whatever you can remember. Okay. So B stands for Barford test, O stands for Osajon test, M stands for Mollis test, B stands for Benedict test, iodine test, Selinov test, Pelling test, and Sucrase test. I'm just giving you the a brief overview of this test. So we will discuss one by one. Okay, please write it down. We will discuss one by one. So first important test of today's discussion is Mollis test. So Mollis test is one of the common tests for the identification of all type of carbohydrate. If I will just explain you about Mollis test, when carbohydrate or monosaccharides when react with concentrated sulfuric acid, it will eliminate three water molecules due to dehydration step. Okay, three water molecules to form hydroxymethyl furfural. Okay, to form hydroxymethyl furfural. Now this hydroxymethyl furfural again in the presence of concentrated sulfuric acid, again dehydration. Okay, again dehydration water leads to what? Furfural. Furfural. Now this furfural reacts with alpha naphthol. Now this furfural react with alpha naphthol, so that will leads to a red color will develop. That will develop leads to red color, purple red color at develop. What you will get? You will get a red color or purple red color. So this test for this test is for what? For the identification of all type of carbohydrate. This Mollis test is for the all type of carbohydrate. Please note it down about Mollis test. Okay. So Mollis test alpha naphthol plus ethanol. So purple red at ring junction for the all carbohydrate. Next is two important tests for ketose. You know there are two different type of tests for the ketose. So ketose means what? Those who have ketone functional group. Okay. Example is your fructose. And from many times, this test question has been asked. So first test is your Selinov test. Second test is what your second test is your Fulgens test. Selinov test and Fulgens test. So in Selinov test, what happens? So resocinol react with in the presence of resocinol in hydrochloric acid. Okay, in Selenov test, resocinol in hydrochloric acid. In Fuelgen test, furfural react with urea in the presence of stannous chloride. So in Fuelgen test, what happened? Furfural plus your ketones in the presence of urea and stannous chloride. Both the tests are for which compound? Both the tests for the identification of ketones. So ketose example is your fructose. Okay, you can see over here. So in Fuelgen test, there is a blue color developed. This is for the detection of ketose. Similarly, Selenov test, the red color developed. It is for the identification of ketones. Okay, it basically distinguishes from aldose to ketose. Basically, this test, many times this question I've asked. So this is the Selenov test is identification of ketose or fructose. In exam, the in question option might be ketose or maybe fructose. You remember this test? Many times this question I've asked. I will show you what are the questions that has been asked in previous year exam. Next is sucrose hydrolase test. So you know, so sucrose is a, yes, reducing sugar or non-reducing sugar? Yes, who can answer? Sucrose is a non-reducing sugar. Okay, so one question I can ask you that sucrose is basically made from glucose and fructose. All of you know about it. Sucrose is made from glucose and fructose. It is made from glucose and fructose. So glucose is reducing sugar, fructose is also reducing sugar. But why sucrose is not a reducing sugar? Yes, who can answer this question? Please answer your question in the comment box. So, you know, glucose is a reducing sugar, fructose is also a reducing sugar. When sucrose is made up of glucose and fructose, then fructose became, sorry, so sucrose became non-reducing sugar. Why? Who can answer this question? This is because when glucose and fructose basically combines with, or glucose is basically aldose. Fructose is basically ketose. Let me explain you in a blank page. So sucrose is basically non-reducing sugar. Sucrose is basically non-reducing sugar. It is of two types. Either this, it is made up of glucose and fructose. So glucose is a aldose. That means glucose have free aldehyde group. Okay, it has free aldehyde group, and fructose is a ketose. It is having free ketone group. When glucose and fructose combine, so this free aldehyde of glucose and fructose are basically combined to form sucrose. So here the reducing, because you know na reducing sugar have free aldehyde, free aldehyde or ketone. So here the aldehyde and ketone is not free right now. That's why sucrose is considered as a non-reducing sugar. So that's why to hydrolyze the sucrose is basically a difficult task. So how we can hydrolyze? We can hydrolyze by we can hydrolyze the sucrose by concentrated hydrochloric acid. Please remember, it's hydrolysis is difficult, so that's why we use concentrated hydrochloric acid for the hydrolysis of sucrose. So when it undergo hydrolysis, it forms glucose and fructose. Okay, so this test is called as sucrose hydrolysis test. This test is called sucrose hydrolysis test. Now, four tests we have discussed: Mollis test, Fauger's test, Selinov test, then sucrose hydrolase test. Next is for the Osazone test. Let's discuss Osazone test. Okay, let me take another white page. Osazone test. When phenyl hydrogen, when phenyl hydrogen react with sodium acetate plus sugar, boils for 2 minutes, then what happens? Osazone form. Glucose, fructose, this will form needle-shaped Osazone. Glucose, fructose, mannose form needle-shaped Osazone. Maltose form sunflower. Maltose form sunflower-shaped Osazone. Lactose form pins or hedgehog crystals appear like a ball of prickles. Okay, please remember phenyl hydrogen, sodium acetate, acetic acid, water heated for 20 minutes, greenish yellow or needle chromostic shaped crystal is formed for glucose, fructose, mannose, lactose form hedgehog crystals, and maltose forms a sunflower like crystal. Okay, maltose form a sunflower like crystals. Okay, I hope everyone is clear up to this, no doubt at all. Next important test is iodine test. So iodine test, this is basically when polysaccharides or solution of like polysaccharides basically react with 1 or 2 drops of iodine. It will form two agents. Either it will form starch or it will form glycogen. Starch is the storage form of carbohydrate in plant. Glycogen is the storage form of carbohydrate or glucose in animal. So if starch is there, then deep blue color will form. If starch is there, then deep blue color is formed. If glycogen is there, then brown color will develop. Okay, please remember many times this question have asked from this iodine test. So when suspension or 1 to 2 drop of iodine react with a polysaccharide, it will give either starch or glycogen. If starch is formed, then deep blue color will be appear. If glycogen is there, then brown color will be appear. This is for the iodine test. Next important three test is for the reducing sugar. Next three test is for the reducing sugar. So what is reducing sugar? Reducing sugars are those which have free aldehyde or ketone group for the reduction. Free aldehyde or ketone group for reduction. Okay, so there are three important tests you should remember for the identification of reducing sugar. What are they? One is your Benedict's test. Next is your Barford test. Another one is your Felling's test. This Benedict test and Barford test for the reducing sugar. Barford test is for identification of monosaccharide. Distinguish between monosaccharide or disaccharide. What is Barford test? When neutral copper red. When neutral copper red react with acetic acid in boiled water bath. Okay. So what it is telling that Benedict test, Barford test. So Barford test is when neutral copper red react with acetic acid in water bath. So what will happen? This will produce copper. Okay, this copper ion react with monosaccharide. Okay, this free copper ion basically what it do? It reacts with the monosaccharides. Okay, when it reacts with the monosaccharide, what it will form? It will form a red color precipitate, and this red color develop within 5 minutes. Okay, if this red color is developed within 5 minutes, then we call it as a monosaccharide. Because if disaccharide is there, it will take more time, to develop color. It will take more time to develop color. Please note it down. So what we are saying that this Benedict test, what is this Benedict test? So Benedict test is, sorry, this Benedict test is telling that uh, this Barford test is telling that when neutral copper acetate react with acetic acid in boiled water, there is a development of red color precipitate at the bottom of the test tube. This is for the identification of monosaccharide because this copper ion basically react with the monosaccharide very fastly. Okay, and the color is basically develops within 5 minutes. Then we call it as a monosaccharide is present. If it take more times to develop color, that means disaccharide there. Benedict test, when copper sulfate react with sodium carbonate and sodium citrate boil for 2 minutes, there is a orange red color precipitate will develop. That is for the reducing sugar. Again, I am repeating, when Benedict test is what? When copper sulfate react with sodium carbonate and sodium citrate in boiling and boil for 2 minutes, there is a orange red color or brick red color precipitate is developed. That is called as reducing sugar. Next is Fehling test. When Fehling solution A and Fehling solution B boil for 2 minutes, yellow red color develop. That is also for the reducing sugar. You can see from this picture, if high amount of moderate to high, if moderate amount of uh, orange red color develop, then we can call it as a moderate amount of sugar is present, 1.5 to 2 gram. If high brick red color precipitate is developed, that means high sugar is present, high level of reduced sugar is present. Okay, this is for the Benedict test result. Please remember. Okay, I hope everyone is clear up to this. So we have discussed what are the point we have discussed. We have talked about Benedict for the reducing sugar. We have talked about Benedict test, Barford test, and Fehling test. We talked about oxygen formation test. We talked about iodine test. We talked about Selenov test. We talked about fuel jet test. We also talked about Mollis test, sucral hydrolysis test. We have discussed all the tests. Okay, I hope no one have any doubt regarding this test. If you have any question or any doubt or any confusion, you can put your question in the chat box or in the comment section. We will answer your question. Okay, now we are going to discuss some of the important MCQ related to this topic. So first question is, this question was asked in GATE 1997. So you have Benedict test, Hayes test, Gemlin test, Salanowski test, and you have to match. So just now we have discussed. So Benedict test is for the reducing sugar. So reducing sugar example is GFML. I gave you one mnemonic, that is glucose, fructose, maltose, and lactose. These are the what? These are basically your reducing sugar. Here glucose is option. So one answer will be U. Next is Hayes test. Why Hayes test is used? Yes, Hayes test is basically used for the identification of bile salt. Gemlin test. Yes, who can answer? Why Gemlin test is used? Gemlin cell test is used for the bile pigment. Or one another test is also used for other than Gemlin test, that is your 𝙂𝙎𝑇's test. Okay, 𝙂𝙎𝑇's test also used for this. And Salanowski test is for the, yes, cholesterol. Selenov test is something different. Okay, Salanowski test is also different. So do not confuse with Selenov and Salanowski. Okay, moving to the next question. Of here, option A will be the correct answer. Okay, moving to the next question. Which of the following test is used specially for the identification of keto sugar? I told you this question we have a potency to ask in exam. So keto sugar means example is your fructose. Okay, keto sugar, which test is used? Just now we discussed. We discussed about Selin-Wolf test. Okay, so Selin-Wolf test is for you. Barcode test, this is for the identification of monosaccharides. Okay, now next question. Uh, Option B is the correct answer. Next question. Distinguish the taste between monosaccharide and disaccharide. Yes, just now we have discussed. So Barford test. Okay, Barford test. So what was that principle? When neutral copper rate react with acetic acid boiled in, yes, boiled in water. So what it will do? It will form, yes, it will react with the monosaccharide and develop a red color. If the red color is developed within 5 minutes, then we call it as a monosaccharide. If disaccharide is there, then it will take longer time to develop. Option C is the correct answer. Next question. Which of the following is a analytical reagent used for the quantitative determination of ketones? Quantitative determination of ketones. Just now we have discussed quantitative ketose or fructose. Which test? Selidov test. Okay. Which test? Selidov test. Mollis test. This is a common test for carbohydrate. This is a common test for carbohydrate. Please remember, this is a common test for carbohydrate. Next question. General test for identify carbohydrate. Dekho. Just now I told you, same question have asked in exam. So general test to identify carbohydrate is Mollis test. Okay. Mollis test is used for the identification of carbohydrate. That is a general test for carbohydrate. Okay, so today we have discussed the detail of all the important tests that is performed for the identification of carbohydrate along with the mnemonic that is bomb which fall safely. So from that name you can remember all the tests for the carbohydrate, and in that test we have discussed which test is for the it is like Selenoid test and fusion test for the specially for the ketose, your Barford test for the distinguish between monosaccharide and monosaccharide disaccharide, Benedict test and Felic test for the identification of reducing sugar, Osajon test for the detection of osajon, sucralose hydrolysis test for the identification of sucrose, iodine test for the distinguish between starch and glycogen. Okay. I hope this session was helpful for all of you. You will never forget about the detailed test of carbohydrate. Which any time in any exam, if such type of question will ask, I hope I am so confident that you will able to answer that question. Thank you. Thank you so much. Stay connected with GDC. Thank you.